and I expect the same vigor of discussion from you. I remember you because I know that Reem is there in the class and she discusses. So I expect the same. Okay. Where were you? On a holiday? On a long holiday?
I was in India.
For a month. You were in India. Okay.
Yeah.
Great.
Yeah.
You enjoyed India?
Yeah.
You belong to here only, right? You have.
Yeah.
Okay. Great. Okay. Welcome back. So
Thank you,
Reem. So Maimuna, how about you? Giving a quick summary. Am I audible? Maimuna? Or anyone who would like to volunteer? Maimuna, Minashi, Alia. Any three of you. See, someone has to give a summary, right? We cannot move forward without that. Come on, I'll help you.
I would, but I was absent in last class, so.
That's even great, Alia. That's amazing of you. Thank you so much.
Yes, sir. I'm not like.
Okay, you will not. You
Yeah,
okay. You're saying you would if you were present.
Yeah. So, sorry.
Okay,
sir.
so you're just marking yourself in my good books. You are already. All of you are in my good books. Don't worry. But just like I have to nudge you a little before I can make you do anything. Let's see who has messaged in the chat. Okay, so Mamuna's internet is fluctuating. My voice is breaking for her. Is my voice breaking for others as well?
No, sir.
No? Okay, then it's Mamuna's network only. Then we are left with Minashi. Minashi, would you save the day for us? Yes, see, Minashi comes as a savior at the last. She waits. Okay, let's begin.
So last class we started with allergies. Allergies are defined as the exaggerated response of the immune system when the antigens enters our body. Then the substance that cause allergies are called allergens. The antibody which is produced against the allergens are called IgE type. The symptoms include sneezing, wheezing, coughing, watery eyes, skin rashes, etc. Then
so it can differ. Just 1 second, Minashi. So these symptoms can differ from person to person and from allergy to allergy. So I was giving an example of my own case where I have allergy towards conditioned air. So I mostly sneeze, wheeze, and sometimes the eyes gets watery. But skin rashes, coughing, difficulty in breathing is not there. But someone who's allergic to pollen, let's say, might have difficulty in breathing. And someone allergic to something that you can eat or drink like alcohol gets skin rashes all around their body. So it differs. Okay, it's not like all these symptoms are present in all the allergies. Yes, continue.
And the cause of allergy is when the allergens enters our body, release of histamine and serotonin when the mast cell is released. Then the treatment includes using the drugs like antihistamine and.
So did you say that mast cell is released? So mast cells are one type of big secretory cells. Yes, from the mast cells, histamine and serotonin is released that causes allergy. Let me tell you, serotonin and even histamine might be released from other cell types as well. For example, neurons secrete serotonin, I told you in the last class, but that will not lead to allergy, right? There it's acting as a neurotransmitter. Okay, a signaling molecule. So it also depends which cells are responding to the allergen. Mast cells mainly do and secrete histamine and serotonin and causes allergy. So the medicines that we take against allergies contains antihistamines. The most common example is Allegra or cetirizine. They are antihistamines to suppress the allergy. Okay, continue.
Yes, then we studied about autoimmunity disorders. It is a condition where the body attack its own cells. The examples are rheumatoid arthritis.
Arthritis.
Yeah.
Okay. Yeah. Rheumatoid arthritis is autoimmune disease.
Then immune system in humans, it consists of lymphoid organs, the primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs consist of bone marrow and thymus.
What are
and
lymphoid organs? Why we call them lymphoid organs? Because
limbs are formed.
No, they secrete lymphocytes.
Yeah.
They produce, or sorry, secretes, they produce lymphocytes, B lymphocytes and T lymphocytes, right? So that's why they're called lymphoid organs. Yes, continue.
So, and the secondary lymphoid organs contains spleen, lymph nodes, tonsils, small intestines. Yeah.
Yeah, and even appendix.
Yeah.
Is also like we treat it as vestigial organ, but even in our ancestors and even in early during the life, appendix is a reservoir for lymphocytes. Okay, then
bone marrow. Bone marrow produce all the blood cells, including the white blood cells, WBC. It is a primary lymphocytes. Then another primary
lymphoid organ.
Lymphoid.
Yeah, bone marrow is the primary lymphoid organ. Lymphocytes are B and T lymphocytes. Yes. Then
the second one is thymus. It is a lobed organ which is located near the heart. Then during the time of birth, it is large in size, but it reduces the size with the ages, and during at the puberty, it reaches significant. It is significantly reduced. I mean, the size is significantly reduced. Then, the secondary lymphocyte organs, the spleen. It is a P-shaped organ, and it has large reservoirs of RBCs.
Okay.
Yeah. Then MALT, mucosa associated lymphoid tissue. It produce B and T cells. It is located.
Yeah, MALT is and just let me make it clear, Minashi. So for those who are not present, and especially Reem, so MALT is a common name given to all those lymphoid tissues which are located, which are scattered throughout our body, but they are not one organ. They are not in one organ, but they are all capable of producing lymphocytes or storing lymphocytes. So they are located in the mucosal lining, okay, of the major tracts, all the tracts that we have. What tracts do our body has? Respiratory tract, digestive tract, and excretory tract, which we call urogenital tract, urinary and reproductive tract combined, and gastrointestinal tract, which is the digestive tract combined, and respiratory tract. These are the tracts. Tracts are basically passages, tunnels, right, everyone? So the mucosal lining of these tracts contains tissues which are scattered, present in pouches known as MALT. So mucosa associated lymphoid tissue, MALT. And as I told you, there is a primary lymphoid organ, which is bone marrow and thymus. Then there are secondary lymphoid organs, which are these 5: appendix, spleen, lymph nodes, tonsils, small intestine. All of them, primary and secondary combined, make around 50% only of our lymphoid tissues. The rest 50% is MALT. Okay, but we do not study it as one because it's not present in any one location. Okay, is it clear, everyone? It's scattered throughout the body. Reem, is it clear for you?
Yes, sir.
Okay, very good. Yes, Minashi.
Then AIDS, acute immunodeficiency syndrome. It is caused by HIV virus, human immunodeficiency virus. It is a deficiency of immune system. It is first reported in 1981. Then these viruses are capable of doing reverse transcriptions. The transmissions of HIV is by sexual contacts or by sharing the blades or razors, then from an infected mother to the fetus through placenta or through blood transfusions. Yeah.
Yes. Right. So there are many reasons through which, many modes through which HIV can get transmitted from one person to another, mostly from one infected person to another infected person. So the taboo that it is only it's only associated with, unprotected or unnatural sex is wrong, and we should not treat the patients, we should not humiliate or insult the patients on the basis of this. Okay, so I want all of you to open your textbooks and go to page number 155. Can you do that? Everyone has an NCERT? I'll also try to pull that figure out for you. Give me one moment. Please open page 155, everyone. Do you find the flow chart showing the replication of the retroviruses?
Yes.
Yeah. Okay, I just want that figure. Where is that? Okay. This is the figure. Okay. So today we are going to do the mechanism of it. So can you all see the figure that I have inserted here? You all can see this?
Yes, sir.
Okay, very well. So let's see what's happening here. Okay, so this is a retrovirus, in which case you can also, let's say this is the HIV, human immunodeficiency virus, which is a retrovirus. Retrovirus means, why do we call it retrovirus? The word retro means old, right? Old kind of life or old kind of viruses. What are the old kind of viruses? Those who were present in the last class should say. What do we mean by old kind of viruses? Viruses which have RNA as their genetic material and not DNA. Okay. Remember the RNA world hypothesis in evolution? People, I hope I'm audible to all of you, right?
Yes, sir.
So viruses with RNA genome are known as retroviruses. Okay. So they have a RNA core. This is the RNA, let's say. And then this is the viral coat protein. So they simply just have two things: a coat protein made up of a coat made up of protein, which is true for all the viruses, and genetic material inside, naked genetic material, which is RNA in this case. By naked I mean not surrounded by any kind of a cytoplasm or a membrane or any protein or anything. Now this viral core RNA infects a normal cell. Now this is a animal cell that they have shown you. This is a normal cell. This is the boundary of the cell, and this is the nucleus. So how it infects? Can you see here what's happening from this step to this step? Can anyone tell me what can you see happening? This is the protein coat of the virus, and this is the RNA of the virus. What do you see is happening when they're infecting the animal cell?
That's when it enters the animal cell. The protecting.
Yes, exactly. What is entering the animal cell? Do you see the protein coat anywhere entering the animal cell?
No, it's just that retrovirus. only retrovirus, not that protein code.
No, but retrovirus has two things. Retrovirus is the whole virus. It has two components, a covering and a RNA sitting inside. Right? Retrovirus means both the protein coat and the RNA together. But here, this virus, so RNA and protein coat together make the virus. Is it clear, Alia?
Yes, sir.
Yeah. But what do you think? What do you see is entering inside in this diagram?
Just the RNA.
Yes, just the viral RNA is introduced into the cell. So where does the protein go? We don't care where does it go, right? It does not enter the cell. Simple. Okay.
Yes, sir.
Because it's not required. Why it's not required? Can anyone tell me? But see, here you can see that what is coming out of the cell. A full virus, right?
Yes, sir.
With a protein coat. Protein coat did not enter, but is exiting out. How is that possible? I'm giving you a hint. Central dogma of biology. What is central dogma in biology? Central dogma. DNA makes RNA.
DNA makes RNA. RNA makes protein.
RNA makes
Protein.
proteins. So here, what part they are following? Here, what's happening in this case? This protein coat is protein, right? And the virus is sending RNA inside the cell. And it has to make protein. Can it just simply make protein in our cells? Technically it can, right? But see, look at the step here. It says the viral RNA is introduced in the cell. So which means the virus injects its RNA into the cell. The HIV virus injects its RNA into our cell. And then that viral DNA is produced by reverse transcriptase. It is making DNA from its own RNA. And then that viral DNA, so this is the step 1. Step 2, then viral DNA incorporates into the host genome. So the virus DNA goes into our nucleus and becomes a part of our own DNA. Why is it doing all this kind of circus? It is a RNA. Why not make proteins? Anyone? Any idea? It is a viral RNA, right? First thing, a viral RNA is not very stable in our cell. It will be degraded. So the RNA which is entering, this RNA will be degraded by our defense mechanism very quickly after some time. Sooner or later, we will figure it out and it will degrade it. So what it needs to do, it needs to make a DNA, put it in our genome so that we do not know that there is a mole among us. And when our cell do the transcription from DNA to RNA, what do we call it? Transcription. Then we will make multiple copies of the RNA again now. So new viral RNA is produced by the infected cell, and this viral RNA is multiple. They are showing you just one, but they will be into millions, let's say. And now what will happen? This RNA will also have all the information to make what? The protein code, right? So new viruses are produced from the protein. So from RNA, now translation will happen because it was the viral. So let me go through the series. It was the viral RNA. So first it will convert into DNA. This is the step 1, reverse transcription. Then this DNA will be converted back to RNA, the viral RNA. Then it will be made into viral proteins and the virus will release. Make sense why it is going back one step? So that it can make millions of copies of itself. Everyone, does this make sense to you?
Yes.
Yes, sir.
And then this cell dies and these viruses, millions of them, release out to now infect other cells. Okay.
Yes.
In many cases, the viruses are very small and this cell can still live while they are being produced and they are being released. So they die later on when the organism dies or when the infection becomes very, very severe. Okay. Some infections instantly start killing the cells. Remember I told you about interferons in this chapter very early on. What were interferons? Minashi,
Yes, sir.
what were
The virus
interferons?
infects the protein secreted by the
Virus protein?
Yeah, the protein secreted by the virus.
No, protein secreted by the cell which is infected by the virus. The cell of our body which is infected by the virus secretes a protein which is called interferon. It helps the other cells in the vicinity, the nearby neighboring cells, to protect themselves. So it is like a signal. I am going to die, you save yourselves, don't die, I'm telling you. It's like that one watchdog or the watchman who's at the front and can see the enemy coming. So he knows that he's not going to survive, but he signals the others, right, that the enemy has invaded. Do you understand, everyone?
Yes, sir.
Don't forget information so fast, okay? Keep revising. So in that case, sometimes the cell dies and signals others. In HIV, it is called very slow infection. So viruses, in HIV, people sometimes can be infected but not show any symptom. This also happened in the case of COVID infection. Some people were COVID positive but were not showing any symptoms. You must have heard or read or seen, right? Some had just the fever, but some were very serious and many died also. So there was a graded response towards the same virus. One reason was our immunity. Everyone's baseline immunity is different. The other reason was the way virus behaves. Okay, so viruses sometimes can stay hidden. They can just multiply, but not produce toxins or not create symptoms. So that's called the latency period of the infection. What it is called? Tell me. Latency period. You understand, everyone?
Yes, sir.
In case of HIV, the latency period can be very, very long. In few cases, it has been reported to be as long as 20 years. So the human was infected with HIV. The human was HIV positive, which means so the infection with HIV may or may not lead to AIDS just suddenly. So the person was HIV positive, but not showing any symptoms of AIDS, was just living normal. And after that, the symptoms started coming. So the latency period for HIV is very, very long. Is it clear, all of you?
Yes, sir.
Okay. So that's why the HIV/AIDS infected person, because this virus does not spread through respiration, does not spread through sweat, tears, or physical contact. It only spreads through body fluid exchange. So HIV can only spread through blood. It can spread through semen, like which are the sexual fluids, and it cannot spread through tears, sweats, or even saliva. Like saliva can spread HIV, let me tell you, but to to spread HIV through saliva in number which can affect you, someone has to drink a bucket full of saliva from the infected person to get HIV positive, which is not possible. Okay, so merely sharing food with a person, drinking water from the same glass, kissing the person will not spread HIV. Is it clear?
Yes, sir.
Is it clear, everyone?
Yes.
Okay, very good. Sometimes saliva, in cases where they say that there were high amount of viruses found in saliva in case of HIV, is because the patient must be having a bleeding gum issue. So now you can understand, if the patient have a cut in the mouth or has bleeding gums, then it's not just the saliva which is there in the mouth. What is also present with saliva?
Blood.
Blood. And we know that in blood the concentration of the virus is more, so that then it can spread. You understand, everyone?
Yes, sir.
Yeah, but anyways, you have any healthy person has to get the virus inside their blood from an infected person's blood, which can also happen just through sharing needles or syringes or razors blades. You should not reuse razor blades while shaving. Okay, so just look at this figure for a moment and let me know if everything is clear to you, or do you want me to repeat something? If not, then I'll move forward and dictate you. So in this case, the cell which HIV, so if we are taking an example of HIV, then this cell in which it is entering is called helper T cells. HIV attacks the T cells of the immune system. There were two kinds of lymphocytes, I told you. So the helper T cells are attacked by HIV. Okay. Is this clear? Shall I dictate things? Anyone has any doubts?
No, sir.
No. Okay. So write down. HIV affects the helper T cells. HIV affects the helper T cells. HIV attacks the helper T cells. And the viral RNA enters these cells. The viral RNA enters these cells. It synthesizes viral DNA. It synthesizes viral DNA through reverse transcription. And the enzyme is called reverse transcriptase. Ase is the word used for enzymes, okay, as a suffix. The enzyme is called reverse transcriptase. This viral DNA gets incorporated. This viral DNA gets incorporated into the host DNA. Host means humans, we. Into the host DNA. And next is new viral RNA is transcribed. New viral RNA is transcribed in the host cell. In the host cell. Next. These viral RNAs are translated. These viral RNAs are translated to produce the viral proteins. Everything that is of virus, we'll use the word viral with it to distinguish. To produce viral proteins. Last sentence, the viral proteins assemble with the viral RNA to make a new virus. So everything gets assembled. All the parts are made separately and then it gets assembled to become a new virus, which is released from the cell. Which is released from the cell. Okay. Now this making of the virus in case of HIV in humans is also happening in the macrophages. So before helper T cells, there are macrophages. Remember I told you what are macrophages? Macro means big. Phages means the cells which can eat up. They are present in our tissues and they just keep clearing anything foreign, keep eating anything foreign which comes in our body. Now this HIV virus use this cell only because it's a very big cell. It can act as a factory. So this making up of new virus particles is happening in macrophages. And simultaneously, they are also infecting the helper T cells. So helper T cells will keep dying. Okay. They attacks the helper T cells. And because of this, helper T cells number decrease in the body. When this decrease starts happening, so now do you understand the reason for latency? If the virus just enters the macrophage and makes its copies and do not kill the helper T cells, it is the person is still positive for HIV, but is not showing any symptoms of the disease. Make sense to you, all of you?
Yes, sir.
Yeah. The moment it starts killing the helper T cells, that is where the helper T cells will start decreasing. And because helper T cells are a very important immune cells, then what will happen as a result?
Symptoms will start showing up.
What symptoms? What will happen in the body? If helper T cells are no longer present, what will happen?
B cells will be not formed.
First thing is, yes, you're right, but it's not like B cells will be not formed. See, B cells are forming from a different place, bone marrow.
Yeah,
B cells will be, it helps the B cells to secrete antibodies. So antibody production will be affected, yes?
Yes, sir.
As a result, what will be affected? What will happen? As a result, our immunity will go down. Am I correct?
Yes, sir.
And what is the name of the disease AIDS? Full form?
Acquired
Immunodeficiency syndrome.
So now you understand where does the immunodeficiency happening from? Because our helper T cells are dying and our antibody production is affected. So our immune system is deficient. That's why this disease is called acquired immunodeficiency syndrome. Now, our guard is gone. We cannot protect our body from many pathogens. So because of HIV infection, any other infection can happen in our body. It can be pneumonia, it can be hepatitis, it can be kidney infection, it can be any other gut infection, it can be gastritis. Any kind of infection can happen in our body, and people often in HIV AIDS die due to other infections like lung infections, kidney infections, liver infections, etc. Is it clear, everyone?
Yes, sir.
Yes, that's why it's called a syndrome. It's a amalgam of anything, like where the symptoms are not very clear what will happen. Anything can happen because our immune system is compromised. Now write down, there is a time gap between the infection and appearance of symptoms in AIDS. There is always a time gap. Now that time gap for some people, if they are unfortunate, can be a few weeks. For some it can be months, and for many it can be years, even like 10 years, and in a few reported cases it has been 20 years as well. But they will come, they will come towards the old age. If someone is middle-aged when they acquired the virus, it will come faster, the symptoms. If someone is young in the 20s and acquired the virus, maybe it can show no symptoms till the middle age, but symptoms will come in the due time. It's not like symptoms will never come. Because anyways, when we grow old, our immune system becomes weak. You know that? Old people are more susceptible to infections. The whole world was trying to save their oldies during the COVID. Correct?
Yes, sir.
Yep. Okay. So this is AIDS. Now write down certain diagnostic assays. How do we diagnose? Any idea? How do we diagnose AIDS? How do we diagnose COVID? What is COVID? How do we diagnose COVID?
Due to symptoms.
Okay, of course due to symptoms, but that's the diagnosis part, like where you are sure that it is the COVID virus. Similar symptoms can be shown by many other viruses, no? Before COVID, what was new in COVID? Nothing was new in COVID. It was just a mixture of two things. Your lungs get filled with water, that happens in pneumonia also. You get fever, that happens in almost every infection. You get sore throat, you lose your taste. All these things, all these symptoms, it's not like other viruses do not do this. They do. So to be sure that you have COVID, what do you need to do? What do you need to test?
RT-PCR.
RT-PCR, perfect. What are we checking for in RT-PCR? What are we looking for? Any idea? We are looking for virus specific genes, something that is only present in COVID. And if that thing we can detect in our body, we can say that, oh, the COVID virus has infected us, the body. Make sense? There will be something, some part of the DNA or some gene that is just the COVID gene, right? The characteristic feature of COVID. Yes or no? Similarly, in case of AIDS, the widely used diagnostic tool is called ELISA. ELISA. Full form is enzyme linked, write down, enzyme linked immunosorbent assay. If you do not remember the full form, it's fine. You just need to remember ELISA. Okay. Now, as the name suggests, what are we looking here for? Enzyme linked immunosorbent assay. So though this is not an integral part of your syllabus, but you should know what's happening here. So what is ELISA? It's an assay. You can do ELISA for many tests, even for many different kind of diseases. or many different kind of viral infections, you can do ELISA. So what are we testing in ELISA? We can also do ELISA for hepatitis B, let me tell you, because that's also caused by hepatitis B virus or hepatitis C virus or HIV, human immunodeficiency virus. So what we do in it is, with the help of an antibody, we are detecting, it's an immunoassay. We are using antibodies to detect certain viral proteins. Do you understand, everyone? If there is a virus in our body, it is secreting it. There will be some virus specific proteins secreted. Yes or no? So if there is HIV virus in any infected person's body, it is replicating, and then it is the RNA is being translated into protein. So if from a sample I can find that virus protein, can I say that this virus has infected the cell? Otherwise, where will that protein come from in our body, right? It is not our protein. Does it make sense?
Yes, sir.
Yeah. So it's a very basic technique, ELISA. Okay, it's done in for many kind of infections. So just write down one line. ELISA is a basic assay technique. It's a basic assay technique which can be used to detect, and measure the concentration of peptides and proteins and antibodies in the blood. Peptides, proteins, and antibodies in the blood. Now, depending on which organism you want to look for, you can make a ELISA test for that organism, for that pathogen. So here we do the ELISA to detect the proteins of a HIV virus. Okay, make sense?
Yes, sir. Sir,
Yeah.
can you repeat the line?
Yes, sorry.
Can you repeat it again, that definition?
Yes, for ELISA, right? It is a basic assay technique. ELISA is a basic assay technique that is used to detect and measure. Peptides, proteins, and antibodies in the blood. Okay.
Yes, sir. Thank you.
Peptides, proteins, and antibodies. Now it works on the simple principle. You can just add one more line. ELISA works on the principle of antibody antigen binding. Sorry, specific antibody antigen binding. This is the principle. We know that one type of antibody can only bind to its antigen and no other thing, right? Yes or no? So this is the principle that we use. So if I have to test that if this kind of protein is present in the blood or not, I will make a specific antibody against that protein and see if it is binding to something by a binding assay. If it does, then I'm sure that this protein is present in my blood, and I'm also sure that this protein can only come from this virus. So hence we are infected by the virus. So it is based on the principle of specific antibody antigen binding. Now that antigen can be a protein also. Is it clear, everyone, for HIV?
Yes, sir.
Okay, very good. So important things that you have to remember is what the virus is. It's a retrovirus. It can do reverse transcriptase, and that is exactly what it does in the macrophages and then also in the T cells when it attacks the T cells. And because of the killing of the T cells, our immune system gets weak and antibody production becomes less. And when we cannot do that effectively, then our body becomes very, very susceptible to any infection and any other kind of infection because of a virus, a bacteria, or a fungi can happen in our body. And mostly people who are suffering from AIDS die because of other infections from bacteria or fungi or any other thing. So it's simple. Our immune system is deficient, so we get any infection. Is it clear, everyone?
Yes, sir.
Now let's talk about cure. Do we have any cure for AIDS in treatment? Do we have any treatment for AIDS? So we have no cure till now. Okay. Only prevention. Is it clear? In case of treatment, but if there is no cure, when someone gets AIDS, they are admitted in the hospital, then what are the doctors doing then, right? This must be your question. If we know that there is no cure, what are we doing? We are doing something called symptomatic treatment. You know what is a symptomatic treatment? Where we do not treat the disease, but we help the patient with the symptoms. Let's say there is a virus for which we do not know anything. We have encountered it for the first time. There is no treatment, no medicines available. Like in case of COVID only. Do we have any medicine against COVID? Do you know? Yes, Alia?
No,
Was
sir,
it?
we don't medicine against COVID
We don't
virus.
have a medicine. Yeah, exactly right. We don't have a medicine. So what were we doing to the patients?
We are just treating their symptoms. Like if it's fever, then we are doing,
Yes, right. If it is fever, give them paracetamol. If they have body pain, they give them painkillers. If they have lung infection, put them on ventilator so that they can breathe artificially, right? If their oxygen level falls down, put them on oxygen ventilators, things like that. So these are called symptomatic treatments. We are just trying to get rid of the symptoms and make the patient survive and fight. So there's no cure. Now, if there is only prevention, how can you prevent? So everything that we have studied above, because of which HIV spreads, don't do that. Like let's go up and keep writing. So we know that transmission happens through sexual contact or infection. How can you prevent that from happening? By using physical barrier method known as condoms. Condoms are the only physical barrier method, only contraceptives that can protect you against HIV infection. Nothing else, no other barrier, no chemical, no physical barrier can protect. Make sense? Do you understand?
Yes, sir.
So by using condoms and by avoiding multiple partners. Okay, that is one thing. Second, through blood transfusion and contact with the blood. How can we prevent that? Keep writing. By making sure that the blood transfusion is happening from a properly tested blood. So when you donate blood to a blood bank, they check for these things. Before giving that blood to anyone else, they check for infections in the blood, specifically for HIV and hepatitis infections, viruses in the blood, and then only it's given to another person. Okay. Also, while transfusion, do not reuse the needles, okay, from one person to another. So needles, everything that pierces into the body, invasive, should be reused. sorry, should not be reused, should be removed, and new things should be used. Third is sharing infected needles, razors, blades. So don't do this. Don't share infected needle. Don't share needles, blades, or razors at all. Simple. So if it is shared between a healthy person, there's no problem. But you never know who's healthy, right? Out in the open, who has the infection and who does not have, you don't know. And even if one person has the infection and that person shared a blood, donated a blood, and that blood without testing went to 10 people, then all 10 of them will have AIDS without a reason, right? So that's not good. Fourth is infected mother to the unborn baby through placenta. Now, can we prevent that? Can something be done in this? This I give you as a homework. Figure out, is it possible to prevent a child, to protect a child if the mother is HIV positive and the mother gets to know later and the baby is already in the womb? Can we still prevent a baby from getting HIV AIDS? Write down this question. This answer you will tell me in the next class, all of you. Okay, Reem? I'm expecting an answer from you.
Okay,
Wafia
sir.
has joined. And others as well. Wafia, how are you? You joined late, Wafia? Am I audible to you, Wafia? Yes, okay. Right, so everyone has written the question. Is it possible to prevent an unborn baby from getting the HIV infection if the mother is infected already? Tell me. Okay, and read the prevention part from the NCERT for that, and you will get some idea. So is HIV AIDS infection clear to you, and how it affects the immune system? That's also clear to you?
Yes, sir.
Okay, now let me begin with one more famous, very well studied disease called cancer. And how is it, why are we reading cancer in immune system? What is a cancer? Anyone knows anything about cancer? I hope you all must be knowing things about cancer. You have also, you know what is cancer, right? Everyone?
It's a deadly disease.
Of course. That's why we are studying about it. AIDS is also a deadly disease. It kills, it might kill slow or fast depending, but at the end it kills. Cancer, they say the same thing, you know. But nowadays, you might also see celebrities, and in the news, you will find people who have defeated cancer. They are proud and they say that we have defeated cancer and they are surviving. We've not heard of these people who have defeated cancer and are surviving,
We did.
right?
Yeah.
So it's not that deadly anymore, or what? We found a cure.
It is, but there are like two types, like benign and malignant. It depends.
So if it is benign, it's not a cancer. You use two words. That's like benign. Yeah, tumors are of two types, right, Alia? You're correct. There can be a benign tumor and there can be a malignant tumor, but benign tumors normally, because they are benign, they do not, malignant means they transfer from one part of the body to the other. They normally move, spread, but benign tumors do not spread to other parts of the body, and therefore they cause very little damage. They still can cause damage and can kill you if there is a tumor in a vital organ, even if it's benign. For example, if there is a tumor in some very important region of the heart, the liver, of the kidneys, or of the brain, it can still, might cause a lot of damage and kill. But if it's possible to remove them, you just get rid of them and they will not spread again. So that's not an issue. It's not a cancer. We can still survive and defeat benign tumors. But in cancers, it's very difficult because it spreads. That's where the deadly part, which I think, who mentioned the deadly? It's a deadly disease. The deadly part comes from its spreading capability. Now, let's write something about cancer first. Write down. It is one of the major cause of death by disease all over the world, and there are many type of cancers. But before going into that, let me tell you, cancer, write down, is the major cause of death by disease all over the world. But what is a cancer? As I already told you, in cancer there are tumors, right? And what are tumors, everyone? Alia, Mamuna?
When cells start to divide, but it doesn't stop and they just divide.
Yes, right. Just keep on dividing. So cells somehow become rogue. They should have stopped at one point, because cells do divide, cells normally keep dividing. Many cells do divide, especially our epithelial cells and skin cells. They keep dividing, but they become cancerous when they stop dividing. But when they stop dividing, they also become tumor. So tumor formation is a important aspect of cancer. Now, why do you think a cell, how do you know a cell know that when to divide and when to stop? Any idea about this? Have you ever thought about it? So let me give you an example, okay? How many of you have got a cut or a wound, a major wound in your life? A major cut in your arms or hand or leg or somewhere. As a kid, you fall and you got a cut. No, I'm not talking about the bruises. Yes, some of you have got. Okay. So when you got the cut, I have got a lot of cuts. I was a very, what do you say? A naughty kid, a very restless kid. So I always ended up in trouble, cut and broken, some part or the other of my body. So if you have seen a wound closely, what is a wound? We miss some certain cells are missed, right? If there is a cut or a wound, certain cells are broken or damaged, right? Right, Wafir? Because you raised the hand, so I'm just directing the question to you. And then what happens over time to heal the wound? What do you think happens? The cells which are missing, what happens to them? Anyone? Even those who have not, very luckily and fortunately, and touch wood, those who have not had any major wound, how do you think major wounds heal? The nearby cells, they divide and fill up the space. Am I correct?
Yes, sir.
So the wound is healed. The cells which were missing now are back. But when they divide, they also know now we have to stop. The wound has healed. How do you think they know? It's very simple. It's logic. If I put 100 people in a dark room, see, cells cannot see each other, let's say. They don't have eyes. So if I put 100 people in a dark room, and the dark room could only accommodate 100 people, everyone, like when they are standing, they can literally be in contact with other people, so they know that there is no space to move here and there. This is how our cells are also packed in the body, right? Now I take 10 people out of the room, so there is some empty space now. People, are you imagining?
Yes, sir.
Now one by one I start adding people, just like our cells are dividing. When will you say that now do not add more people? You are in the dark, you cannot see, but you can speak. So I'm adding people. When will you say to me to stop? Collectively.
When that room gets blank.
So how many people are added back?
10.
10 or 11, let's say. When I will add the 11th person, you will know that there's no space now, right? How will you know that there is no space now? You cannot see. How did you say? What was your why? Someone just give me the answer, no? On the 11th, I will say that to stop. But how? How will you get to know that you have to stop? I have to stop adding people. People, the answer is very simple. Come on. What you're thinking of is the right answer.
There
Just
is
say
no
it.
space to move.
How do you know that there is no space to move? That's what I'm saying. Which senses of yours is helping? Are you smelling that there is no space? You can't see. Are you smelling that there is no space? Are you tasting that there is no space? What sense is helping you?
I think like we are like it's so closely packed that everyone is touching each other. I don't know.
Exactly. See, this is the right answer. Why are you not saying the right answer? It's the touch. You are in contact from all the sides. Both your hands are touching the person who's standing left and right. You can touch, or you can sense the touch from the person standing in front and person standing at the back, let's say. So from all sides, you know that you are in contact with people, which tells you that there is no space. Otherwise, you would have not been in contact from all the sides. Yes or no?
Yes, sir.
Simple. This is what happens. So cells have this property of contact inhibition. The word contact inhibition means when the cells come in contact with each other from all the sides, and when there is no space, then they stop dividing or growing further because they are inhibited by the contact. Make sense?
Yes, sir.
Yes, sir.
So when the wound heals and again it comes back to the normal number of cells, then all the cells again are in contact with, because the layer is complete. Your hand, the lining of your hand, upper epidermis, which got broken, has now been sealed again. So wound is a puncture. You seal the wound, every cell is sealed. So they stop growing then because there is no need. There is nothing left to seal. In case of cancer, this property is lost. So cancerous cell lose the property of contact inhibition. So even if they are packed together, they will still exert force and continue to divide. They don't grow much, but they divide continuously. So in every cell cycle, in class 11th, you must have studied that in a cell cycle, there is a resting phase, right? Cell divides and then rest to grow in size, then again divides, then again grow in size. Yes or no?
Yes, sir.
Here they are just continuously dividing, not growing in size. But still, when they are continuously dividing, they are also growing in size to some extent, but they're not resting as much as a normal cell should have done. But what will happen after some time? If you are constantly pushing against the layer, you will burst the layer, right? And come out as a bunch of tissues, as a tumor. What is a tumor? A bunch of tissues that actually has broken from the lining and come out, right? It's like if you keep pumping air in a balloon, what will happen? It will burst, right? So layers will also burst and that becomes a tumor. Correct, everyone?
Yes, sir.
That is what is cancer, and it's problematic because cancer cells take a lots and lots of energy. You can just understand, to make a tumor, you need lots of energy, right? Now, write down. And there are many types of cancers, okay? In different tissues. It can happen in any tissue. Depending on which tissue it happens in, we name that cancer as lung cancer if it is happening in lungs, blood cancer if it's happening in blood. Okay. Now, blood is the only liquid or fluid tissue in the body. So, anyone knows what is the name for blood cancer? what is the biological name for the disease, blood cancer? It's called leukemia, because leukocytes keep dividing. RBCs never divide, you know that, because they don't have a nucleus. Did I tell you this? RBCs are produced and they die, but they don't divide. WBCs do divide, so they keep on dividing, and become too much in the blood, and that's called blood cancer. Okay, now yes, write down. In the body, cell growth and division. In the body, cell growth and division is highly controlled and regulated. Highly controlled and regulated. But in a cancerous cell, these regulatory mechanisms are faulty. These regulatory mechanisms are faulty. Next line, normal cells show a property. Normal cells show a property of contact inhibition. Normal cells show a property of contact inhibition. Which means contact with other cells, inhibit the uncontrolled growth of a cell. Contact with other cells will inhibit uncontrolled growth of a cell. Cancerous cells have lost this property. Cancerous cells have lost this property. So last line, as a result, the cancerous cells continue to divide and form tumors. Cancerous cells continue to divide and form tumors. Make sense, everyone?
Yes, sir.
Yeah. So no contact inhibition, and because of which tumor is produced. Okay. Now, tumor are of two types. First is called benign tumor, and the other is called malignant tumor. A malignant tumor is called a cancer because cancers can spread. Now, what is a benign tumor? First, write down. Benign tumors remain confined to their tissue of origin. Remain confined to their tissue of origin. They do not spread to other tissues or other organs. Okay. Whereas the malignant tumor show mass proliferation. Proliferation means? What does the word proliferation means, Minashi? Any idea?
Dividing.
Yes, very rapidly dividing and growing in number, okay, in size. So mass proliferation happens and they can invade and spread to nearby tissues. So these cells actually can move from one organ to another through blood or just through like cell migration. So invade and migrate to other tissues and organs. Now this property of them moving or spreading is called metastasis, also known as malignancy. The spreading capability of cancer is called metastasis or malignancy. Okay. Is it clear, everyone?
Yes, sir.
Yep.
Yes, sir.
Now the term given for this kind of cell, a malignant cell, is called neoplastic. So these are neoplastic cells. Neoplastic means actively dividing and proliferating. Okay. Now, because at one point of time, every cell should stop dividing and do the function. They just have one function to continuously keep dividing. So if a system continuously keeps running and dividing, is it energy heavy or not?
It is.
It is, right? They will need lots and lots of glucose and amino acids and every energy, lipids, fats to just make new and new cells. So do you think a cancer in the body is competing with other normal cells? Yeah.
Yeah.
Yeah, of course. And what happens is that because of cancer cells, the other cells, the other normal cells of our body are deficient for many vital nutrients. And that is also how cancers are also diagnosed, you know. How can you detect cancer? Tell me. Now I've given you the properties of cancer. Before I go to the mechanism and everything, let's do the detection part. Tell me, how will you detect a tissue which is actively dividing and have lost the ability of contact inhibition? And you are giving me the answer. Tell me. One is to just simply study there are many ways to detect cancer. Okay. The most common is to simply study the tissue. take a tissue sample if it can, like if it's a skin cancer, if it's a lung cancer, if it's a cancer from where the tissue can be taken. So a very small tube is sent and some amount of tissue is collected. It is called biopsy. Have you heard of the word biopsy?
Yes, sir.
Yes, in biopsy, a small probe, a small tool in a tube is sent in the body. And what it does is it pinches, literally, just like pinches some part of the tissue, which is enough for us to study. We just have to take those tissues and see if there are cancer markers, or if we see that the contact inhibition property or the cell cycle or the division cycle is perturbed, is defected. We know that it's a cancer. First way. So write down, first is cancer can be detected by biopsy and histopathological studies. Histopathological means, pathology means study of disease or disease causing agent. Histo word comes from tissue. So we are studying tissues in biopsy, so we call it histopathology. Okay. So all these labs are called pathology labs, where they figure out if there is a disease or not. So that is one way to study, one way to know that there is cancer. In case of blood cancer, you can simply take the blood and count the number of WBCs. So everyone has a normal range of WBCs, which should be around 5,000 to 10,000, let's say. But if someone has 20,000 WBCs, like per unit of the blood, then it means that the person has leukemia. Yes or no?
Yes, sir.
Yeah. Okay. So in case of blood cancer, second strategy is that. What else can you do?
X-ray.
X-ray. Okay. So how can X-rays tell about cancers? X-ray, you don't think X-ray is used for? I have seen people doing X-ray to figure out fractures in the bones. How can they tell you about cancers? You are right. X-rays are used, but not in the same way, the way we use it to identify bone breakages or fractures. But how do you think X-ray can be used?
Like its intensity is lower, so it doesn't invade the tissue or something like that. And then if there's a big mass, it can be like.
Yeah, so she's right. If you increase the intensity, then it will go through the tissue but not go through the bones. If you decrease the intensity, you can also see the tumors, right? This is what you are saying.
Yes.
Correct? Yeah, that's good. Good, very good. You're correct. That's one way to figure out tumors. Yeah. You can also do CT scan. You know CT, the word CT scan? X-ray, CT, MRI, all these are scans of our body. X-rays, you send a beam of X-rays, they pass through our body, and whatever is the dense tissue, they will not be able to pass through that. They will only be able to pass through they can only pass through the softer tissue. So if you use different intensity, based on that, you can actually figure out what kind of tissues have tumors. It's okay. CT is called computed or computer tomography. Tomography means making sections. So in a CT scan, you must have seen, I think in movies, or I don't know if you have seen it in real, there is a big tunnel in which they send the human on a bed inside. Right?
Yes,
The
sir.
human is sleeping on a thing which goes inside a tunnel and then comes back. So they are making sections, computer sections of per centimeter or per millimeter of the human. It's exactly like slicing a bread, but they're not slicing here for real. They're slicing just for imaging through a beam of um, uh, radiation. Okay. Same as MRI. MRI is called magnetic resonance imaging. This can also help you to see inside the body and to detect any tumor. But basically what are we detecting is there is another way by which you can detect, and that is through all these modes. I told you that these cancer cells they actively utilize glucose, right? Because they are competing for energy. Understood, everyone? So if I send a radio labeled glucose, if I make a person drink a glass of glucose or put a glucose in the blood, which is labeled, radio labeled, then when I do the radio imaging, the radiography, these areas will light up. They will send signals. And I can see that on the screen. So I can know which part of the body is actually utilizing more energy than the normal. From this technique, we can get to know that all the tissues, let's say in the body, in the stomach, are normal, but there is one lump of cells near the liver, which is just glowing up. Simply, this is utilizing more glucose. So maybe that's a cancerous tumor. You understand? Makes sense to you, everyone?
Yes, sir.
But we cannot figure out brain tumors through that. Can anyone tell me why? Through this technique, it's difficult. Can you tell me why?
Can you think that glow thingy in the brain?
Sorry, can we?
We cannot get that glow something like in the brain.
No.
Yeah.
Actually, we will get the whole brain glowing all the time, even if you don't, because brain uses the most glucose, right? Do you know that 20% of the oxygen is alone utilized by the brain, along with a lot of glucose? Make sense?
Yes, sir.
Yeah. Okay, so your homework is that you have to read till page 158. Okay, just the cancer part. Causes of cancer, I'll tell you in the next class. If you do not understand, cancer can be caused by viruses, by a mutation in the genes, can also be caused by other substances like chemicals which are called carcinogens, like nicotine is one carcinogen, by physical carcinogens called UV radiation, etc. So this, just read and finish it from